Good morning. Good morning, everyone. It feels like it was yesterday when we were welcoming you all to Barcelona in La Pedrera, and somehow here we are with the last event. And today it's a very special occasion. We are eager to hear your presentations. We want to know how these 2 weeks have gone. And today I'm not going to take much of your time. Just, I need to say a couple of things. Thank you and congratulations. I want to say congratulations to all the participants who have gone through all the challenges that presented this Barcelona International Youth Science Challenge. And thank you to all the participants for trusting us, for applying, and for completing the program. Thank you also to all my team at Fundació Catalunya La Pedrera. They've been working very hard these 2 weeks, but not only these 2 weeks, they've been working very hard to make all this possible during the whole year. Thank you to all the research centers in Barcelona that year after year want to participate and do it brilliantly. Thank you to all the researchers that have been with you all these days. Thank you to our fellows that have been looking after you all these 2 weeks as staff members. That's something that we are very grateful for them. Thank you to the families and friends that are here today and that put their trust in our organization and send their kids to us. Thank you to all the teachers around the world that believe in our program and year after year also place their confidence and trust in us. And also many and every year more and more organizations that are partnered with us and send us their talent from the original countries to here. Thank you, congratulations, and we are here to listen to you. So, Mar, I think it's your place. Thank you very much.
Thank you for your words, Luis. And now the moment has finally arrived. Over the past few days, each of you has taken on the challenge of becoming a scientist. You have been asking questions, exploring new ideas, working together, solving several problems, and turning your curiosity, this very, very powerful fuel, into exciting projects. And today, right now, is your chance to share everything you have been learning. We are genuinely excited to hear your presentations. We've been looking through the presentations and they look so good, so we're excited to hear you for real and celebrate with you everything you have achieved. So thank you for making these days very, very special. And congratulations on everything you have achieved. We are so proud of you. Now, the stage is yours, so please, let's welcome the first group.
Thank you so much, Mar. Good morning, my name is Olga, and during these past 2 weeks, I have had the pleasure of being the staff member of the amazing team of the Planarian stem cells. And before they come up to the stage, I would like you to give them a big round of applause, because I know that they have worked so hard, and I'm really, really proud of them. So, yeah. Welcome to the stage to Anania, Pau, Nolan, Dani, Trini, Berta, Roxy, Claudia, and Fatima. This is for them.
Hello? Well, as an introduction, Planarian is a small flatworm that has an amazing ability to regenerate their whole body for very small parts. This is thanks to having a great amount of adult stem cells called neoblasts, combined with some genetic regulation and biochemical pathways. Also, this process of regeneration consists of cell differentiation, patterning, and growth.
Planarians have a remarkable ability to restore amputated limbs and regrow entire functional nervous systems. They provide a model for regenerative medicine and provide new ideas for treating spinal cord and other nervous injuries. By patterning their signaling and studying their stem cells, we can open doors and discover more about regenerative medicine, longevity, and healthy aging. Our main question in this project was, how can gene knockdown and irradiation reveal the mechanisms behind Planarian regeneration?
So the experiment 0 consisted on cutting the planarians and studying how they would regenerate. During this past week, we have observed how the blastema and the planarian regenerated depending on how they had been cut.
Okay, so now we are at experiment 1. In this experiment, we observed the evolution of the regeneration of planarians alternated with RNAi, silencing, and irradiation.
For experiment 2, our group learned how to conduct a double immunostaining, where we used fluorescent antibodies to bind to our target proteins, which in this case was PH3, which we used to mark proliferating stem cells in planarians, and 3C11, which was used to highlight the nervous system of planarians. And putting these images under confocal microscope, we could compare between the anatomies of control and problem planarians.
We investigated the effect of irradiation on planarian regeneration. We looked at live planarians, the control, the sublethal dose of radiation and the lethal, and we saw that the blastema didn't form in the lethal and formed completely in the control and only slightly in the sublethal. Also, we marked the proliferating cells, which are the red dots, and those are the cells that are undergoing mitosis. So with this, we could see that in irradiated planarians, the irradiation destroyed all the stem cells and meant that planarians were gonna die.
After comparing the control groups, the control planarians with experimental groups, we were able to identify the functions of the mystery genes. Gene X, later identified as beta-catenin 1, produced planarians with 2 heads and even extra eyes when knocked down, showing that it's essential for establishing the anterior and posterior body axis, therefore being responsible for tail growth. In contrast, gene Y, CBP3, resulted in animals with no eyes and no brain, as we could verify using the fluorescence microscope and immunostaining. However, a blastema was still present, meaning that the number of proliferating stem cells did not change, but these were not able to differentiate due to the absence of the gene. This indicates that CBP3 has a crucial role in planarian cell differentiation.
Okay, this is a process called microneedling, and it allows us to inject a small quantity of liquid at a microscopic level, and it allows us to not only block gene expression, but also it structures the gut and other organs inside the planarian, and it allows us to further improve our study of this organism.
So summarizing, planarians are full of pluripotent stem cells that help them regenerate completely from other parts of their body. So investigating these genes from regeneration, how they are expressed or silenced, can help us get closer to similar mechanisms in humans. But for us, the take-home message is the experience of working with people from all around the world that will always be in our memories. So thank you very much for this opportunity, BIYSC.
So good morning, everyone. I'm Pablo Gomez, the staff member of the Quantum Entanglement group at ICUP. And before, I would like to tell something to my group, which has been amazing. You have done really, really amazing job. We have worked with really complicated stuff, and I'm really, really proud of you. So I would like to applaud them a bit. Finally, I would like to welcome to the stage to Oriol, Naya, Amirali, Sabina, Pau, Pinar, Isidora, Guillem, Jack, Milo, and Goku.
Hello. So first of all, we're going to talk about the main objective of this project, that it's trying to understand quantum entanglement and superposition, which are the core topics of quantum mechanics. But like Richard Feynman once said, no one really understands quantum mechanics. So what we're trying to do here is learning as much as we can to get closer to unraveling the behavior of that's unobservable. So now we're going to do a little skit so you can understand this better. Doctor, please help me.
Are you alright, madam? My name's Bob. I'm a doctor in physics. How may I assist you?
I don't know if my little cat is okay. She has been in that box for days. Can you help me?
Based on the symptoms, it sounds like your cat is a quantum cat.
What? What is that?
Well, it's a bit tricky to understand, but your cat, as long as we don't open the box, is both alive and dead at the same time.
At the same time? How is that possible? I don't understand anything.
It's all right. I can get my colleagues to explain it to you again. Maybe a smaller example can help us understand it from a different perspective.
Yeah. Okay, Grandma. So basically, quantum physics, superposition states that, unlike classical physics, particles can not always be in one definite state. So before measurement, they actually exist in a linear combination of all possible states. It's not that we don't know what state they're in yet, but they're literally in a combination of all states.
So next, I'm going to talk about the Mach-Zehnder interferometer, and it's one of the most popular examples of superposition and a simulation of wave-particle duality, which is the ability for a particle to act as both a wave or as a wave of light or a particle of light. And one of the most important aspects of the Mach-Zehnder interferometer is the beam splitter, which is able to create a superposition between light that passes through the beam splitter and gets reflected off of it.
In a quantum mechanic, photon exists in superposition that divides into two passes until it's measured. When they come back together, the interference creates the dark and bright things like on this photo. And that interference pattern shows us the main idea of the experiment. How quantum superpositions leads us to the wave-like behaving of the light.
Quantum entanglement links the states of two particles, putting them in the same system. This means that the measurement of one determines the state of the other, no matter how far away they are from each other. Niels Bohr believed this was a purely probabilistic process. However, Albert Einstein thought there was a hidden variable that determined which state was measured when.
So Albert Einstein's hidden variable theory can be explained by the ball analogy, which basically is that we have two boxes. We put a basketball in one box and a football in the other one, and we send each box to one to Bob and one to Alice. So we can know what's in both boxes by only opening one, because one state determines the other. But there's a problem here, that we know the state of both boxes before sending them. And that is why Albert Einstein thought this process was deterministic.
So Einstein and Bohr both had their own theories. So a scientist called John Bell theorized the test to find out who was actually right, and who would have guessed that Einstein was wrong. This shows us that everything that happens at a quantum state is completely probabilistic.
the last phase of every experiment like we did is obviously the measurement. As we said before, everything in quantum physics is described with a wave function. That is basically the probability of finding each state, in this case, of the cat. The problem is that when we measure the state, the wave function loses all its quantum properties and collapses in one of the two states. Now the cat has to be in either dead or alive, and we can check that is actually dead.
Einstein once said, "God does not play dice with the universe." However, now we know that our world works quite in a more similar way to this Stephen Hawking's quote. Not only does God play dice with the universe, but he sometimes throws them where they cannot be seen. This project has allowed us to really understand and get a little closer to knowing where these dice are thrown and how our universe works. So thank you very much.
Hello everyone, my name is Mireia Rivero and I'm the reference staff for the group doing their BIYSC in the IRB. So they have worked really hard. I'm really proud of them and I'm sure that you will enjoy their presentation. So please welcome James, Narcís, Lexi, Nina, Celia, Clil, Alonso, Filippo, Maria, Irene, Antonieta, and Raya.
Well, first of all, good morning, everyone. As you can see, we're the Decoding Tumorigenesis project group, and we're here today to offer you a small glimpse into what we've been working on for the past few weeks at the IRB Research Center, using fruit flies as a model for biomedical research on the appearance and development of epithelial tissue tumors.
Okay. Drosophila is commonly used as a model in biomedicine to investigate diseases. So in our project, we use them to understand how the tumor grow and how they affect our organism. So to do so, we use a larval tissue that we call wing disc in order to understand the tumor behavior.
So, why use fruit flies as a model organism? First of all, they have a simple genome of about 4 chromosomes and a very short life cycle of about 10 days. They're very cheap. There's very few ethical concerns in dissecting them. They have a really high fertility rate, and most importantly, 70% of human disease genes are conserved in the Drosophila. And they're cool.
So we achieved the flies with the desired genome for the experiment with the GAL4 UAS system, which has 2 components. The GAL4 gene, which is expressed with an enhancer specific to one tissue in the driver line, and the UAS sequence with the gene that we want to express in the tissue in the responder line. So when we mated a driver line fly with a responder line fly, their offspring would inherit both parts, which together activate the gene in the one specific tissue. And more specifically, in our experiment, we targeted the dorsal and the posterior compartments of the wing disc to express tumor growth.
We can cross groups of flies with different transgenes to produce the offspring with desired genotypes. And we do 3 different types of crosses. We do a control, a tumor, and a rescue group.
For the tumor one, we added a suppressor of the coordinator of cell division. So instead of dividing properly, they start to be aberrant and they create chromosomal instability. And then these aberrant cells normally would die, but we added a component called P35 that blocks cell death. It doesn't allow them to die.
We add P35 to the control group as well. This ensures that the differences we observe between the groups is due to the genes being tested, not P35 itself.
And for the rescue, we added the same things as for the tumor, but we added a blocker of the stress chain reaction that the chromosomal instability creates in order to see if we can avoid the tumor growth by blocking this chain of reaction that chromosomal instability creates.
In order to obtain the wing discs, we first need to dissect the larva by removing the posterior end to access the internal tissue. Afterwards, we invert the larva to remove the fat body and isolate the wing discs for further analysis.
Once we fixed the tissue, we used antibodies to stain the samples. Specifically, we used indirect immunostaining to amplify the signal. Then we used confocal microscopy to visualize these stainings. Since it's a laser-based imaging system, it provides higher magnification and resolution than light microscopy along multiple focal planes. Then we combine these focal planes into one 3D visualization in order to quantify abnormal tumor growth and abnormal protein expression.
So we created a tumor on the posterior part of the wing. That's the part that's not in red. This activates a stress pathway, which we marked in green, that results in tissue overgrowth. In the rescue, we blocked this stress pathway that resulted in the blocking of the tumor growth, and therefore the wing goes back to being normal sized. That's why in both the rescue and the control, the posterior part of the wing represents 40% of its area, whilst in the tumor it represents 60%.
In the Aptilus model, we can see that in the control sample, cells remain their original regions. However, once we go to the tumor sample, we can see an extensive cell migration as a result of a significantly higher expression of MMP1, which is a protein that triggers the JNK signaling pathway, and this drives cell invasion. Thus, when we look at the rescue sample, we added this blocker, right, to inhibit this pathway, which allowed cells to return back to their original regions and inhibit further migration.
In conclusion, studying tumorigenesis helped us understand how healthy cells can become cancerous and how this process can be stopped. By studying model organisms like Drosophila and genetic tools, researchers can uncover treatments that could save millions of lives. Finally, this project gave us appreciation for research and teamwork.
The last thing is that we really enjoyed this project, and thank you everyone for listening.
Okay, so hi everybody. My name is Marc Farrés and I've been the staff member for the synthetic biology group. You guys have done an incredible job and now it's your time to show us all. So let's give a great applause to Pauline, Julia, Sofia, Barbara, Joanna, Jana, the other Jana, Rocio, Namu, and Benji.
Okay, so we're going to start by doing a bit of an introduction. Well, synthetic biology is a growing branch of biotechnology, and it's an interdisciplinary field that combines both engineering and biology to power major innovations in many fields like health, agriculture, or sustainability. Using genes like code, scientists are able to construct and design biological parts and also redesign existing organisms for useful purposes. Our goal with this project was to create a genetic circuit and learn how to tackle real-world problems thanks to this field.
So synthetic biology is important because it's like enables scientists to engineer living organisms to address real life world challenges. If it can improve healthcare, support more sustainable agriculture, and help protect the environment. For example, engineered bacteria can produce strong spider-silk for lightweight protective materials, and scientists can also engineer viruses to target and destroy cancer cells while leaving healthy cells largely unharmed.
Okay, so as Joanna said before, we've been working with something called genetic circuits that are some kind of circuits made with genes and proteins. And so we have this circuit formed by different elements. First up, we have the constitutive promoter, that it's like kind of a start button for that circuit, and it tells the gene, that in this case would be TETR, to be active and be expressed. So as it is constitutive, it's always on, so therefore that gene is always expressed. Then we have another molecule that it's like the TETR but like the upper, and it kind of inhibites the sequence promoter, that is an inducible one that can be on or off. And so if we have that molecule and it inhibites the other promoter, therefore the other gene, it cannot be expressed.
So what is CRISPR? CRISPR is a Nobel Prize-winning gene editing technique. The CRISPR-Cas9 system consists of a Cas9 enzyme that cuts specific sections of the DNA and a guide RNA that guides that enzyme. Researchers use this technique to insert new genes or cut out faulty sections, which helps cure genetic diseases. CRISPR is incredibly versatile. So, for example, in our lab, we use the dead Cas9.
So the dead Cas9 is a type of Cas9 that we learned about. There were various types. So what it does is that it, instead of cutting the gene or the DNA, it binds to it and it still has the normal gene regulation process happening. It just does it in a slightly different way. So we were able to see how it would link to the lab experiments that we did. So as you can see here with the green fluorescent protein in the Petri dishes, we were able to see different combinations of these connections, and we saw with the like genetic circuits how it would link with the other information that we learned. So all of them kind of went hand in hand.
Hi, so we were in charge of the wet lab. Now we're going to walk you through the process from the first step all the way to reconfirmation. So at the start, we have the process of PCR, where we amplify DNA, which is making millions of copies from one single DNA strand. Gibson assembly is where we join all these fragments into one complete plasmid. Then we have DNA purification, is where we discard all the materials that we do not want, leaving only the DNA. Bacterial transformation is where we allow the bacteria to accept our designed plasmid. And bacterial selection, where we select individual colonies.
After this, we have to confirm, obviously, that the colonies have the plasmid, and we do this with electrophoresis. After we are ensured that the colonies have the right plasmid, we have to grow these colonies to make more of the plasmids. Once the bacteria is successfully grown, we extract the plasmid and isolating it, and then we sequence it using a flow cell to make sure it works. And something that was extremely humbling is the fact that a process that took us about a week or so can actually be done in just a few days with a machine.
So you just heard from the wet lab team, which is working in a lab, but there's a big part of synthetic biology, which is working with computers and modelization. So Jana earlier introduced you to a genetic circuit. And what we did is modelize, using different constants, different concentrations, our protein of interest and the concentration that we would have in the end.
Okay, so, go back for just a second. So with that equation that we have there, we were able to create this graph using Python, which on the y-axis has our protein of interest, and along the x-axis has the concentration of the inducer molecules that we learned about before. And when we plugged in our data and using that equation, then this was one of the nicer looking graphs that we got. But we also got graphs, if we could press the button again, that were maybe not as nice or kind of with the model that we created before. But what this kind of shows us is that biology is messy, and you're not always going to get the desired result. And that was kind of a big takeaway that we had from this course.
And sometimes there's a bacterial pandemic and you have no idea why.
So besides all of that, we also got the opportunity to participate in a competition between us. So the name is Mini iGEM, and it was inspired by the real science competition called iGEM. And we were challenged to choose a problem in the world and try to solve it through synthetic biology. So here are some of the projects that we developed. Some are in diseases, others will focus on more environmental issues. But then we constructed some scientific posters, we presented it, and we voted the winner. And our winners were Joanna and Benji.
So these 8 days were pretty long, but also pretty short, I'll be honest. We made a lot of memories, and they're definitely ones that we're gonna keep forever. So these are just some of the photos so you guys can get a glimpse of what we did and what we had.
Overall, this project has given us many challenges, but we've learned that in science, practice makes perfect.
So as you can see with these beautiful artworks, we saw that science isn't solely just like doing experimentation and like always gathering data. You can look at it with different perspectives, and in this case, we looked at it with the perspective of art. So we drew with bacteria in petri dishes and waited for it to grow. And one major takeaway that we took, as Benji said, is that science is very unpredictable. You never know what's going to happen. So to end this, we would like to thank, obviously, Marc, and then our instructors, Carles, Elvira, Maria, Guillermo, and Cristobal, and then obviously the iGEM team helped us a lot with the posters and the process of presentation.
You guys helped us so much. You're amazing mentors. It was so much fun to get to learn from you guys and learn from real professionals. Thank you so much, and thank you, all of you guys, for listening.
Hello, I'm Laia, the staff member for the Cosmic Rays group, and I'm really glad to be presenting them because we've had 2 amazing weeks full of fun and learning, and I'm really proud of them. So please welcome Athena, Sahara, Lara, Mizara, Denise, Victoria, Valeria, Estefan, Omar, Carlos, Roxana, Elena, and Stephanie.
Hi, as far as you know, we have tons of different groups, but it's the first group that is making astrophysics today, and it's the only one. We were detecting muons, it's such unique particles, which you will get to know about more today. And something that's really important that we had a lot of fun, we got to know a lot of things. We're really thankful to everyone who got to impact our time here. And I hope you'll have as much funny moments right now, or you will be just really happy to see all of that as we did.
Okay, first we want to do a bit of introduction about the theory we studied. First of all, what are cosmic rays? Cosmic rays are high energetic charged particles coming from space that travel at nearly the speed of light. They are originated in the sun and other energetic astrophysical sources such as gamma ray bursts, pulsars, and supernovae. When they reach Earth, they hit the atmosphere and they produce a shower of secondary particles, including muons. Muons are fundamental particles similar to electrons, but like 200 times more massive.
And although they live just for 2.2 microseconds, they travel at nearly the speed of light, so time dilation affects them, and they are able to reach Earth. And also, they interact really weakly with matter, so they can pass through a lot of substances.
Well, detecting muons matters because they can pass through dense materials, being incredible useful tools for science and security. Among the reasons are help to search new physics beyond the standard model. They can detect nuclear material and also are essential to discover new particles, and understanding these particles are essential for understand more our universe.
Our main goal was to design a detector that detects muon and be sure that the only particle that is getting detected is the muons. In order to do that, we designed a circuit for our detector, and also we use reflective foil for our plastic insulator to maximize the amount of muon getting detected. And also, we did all the experiments inside of the black box, so we eliminate any lights that might disturb our detector.
Our big question that we're... The big question... Okay. Our big question is how we can detect these rays and muons. Can you move on? Thank you.
So, in order to maximize our muon detection, we had to take into consideration these three things, right? Over here. First up, to reduce the background noise, we put the detectors in a big black box, right, to block out, the light and everything. Then we place the two detectors on top of each other, right? So the probability that a muon passes through is slightly bigger. And then to shield it, we basically put the detectors in another aluminum box. I think it was aluminum, but yeah, to shield it from other like electrons or neutrinos that might pass.
Now we want to find out what are the components that make up this muon detector. The first component is the plastic scintillator. It's a piece of plastic that takes in the light from the photons and produces a sense of electric current that passes on to the silicone photomultiplier. The silicone photomultiplier we place directly onto the plastic scintillator in order for it to take in the electric currents from the photons. And then this small amount of electric current that the photomultiplier produces, we have to put into an amplifier in order to find out how strong or to be able to look at it. And then by the end, we use an oscilloscope to look at the movements and how many muons we detect. Okay. On our board, we place the silicone photomultiplier over there onto the bottom, and we solder and weld everything into place. The components are all connected by connection wires, and there are capacitors placed in parallel, and the amplifier is placed at the end, and the resistors are placed all connected by wires. Now, this is one of our projects. We worked on it. It's called the bomb. And then this is our oscilloscope. If you can look at it, you can see a couple of muons that we've detected, and we also detected one coincidence. We detected a lot of coincidences, but we'll move on to the results later.
So in the oscilloscope video that you saw just now,
we had 2 channels, and if only one channel went up, we didn't put that down as a muon because that we categorized that as background noise. Only the coincidences were counted as muons. So here are the results. In this graph, you can see the signals we got categorized according to their voltage amplitude. And what this graph means is that we got exponentially less number of signals at higher energies. No. And 2 other important results were the flux we calculated, which is 203 muons per minute per meter squared, which essentially means that if you trace out an area of 1 meter per mole meter and you wait for 1 minute, you can get approximately 200 muons if you have a detector as good as ours. And like we mentioned earlier, not all the signals we got were muons. Some of them were background noise caused by thermal photons or electrons or other charged particles that could come through. And when we calculated the ratio between them, we saw that approximately only 35% of the events were coincidences, which means they were muons. And as a conclusion to both the experiment and these 2 whole weeks that we got to spend together, we went through all of these 4 stages, which we saw was equally important. We first learned about the theory of particle physics and how the components of the detector works. There you can see us modeling particles with Legos. Then we got to building and testing, and lastly improving, because the last model of the detector that we took the actual data with wasn't actually the same as the first 4 models we developed. And also we had a lot of fun in between. We want to thank all of you.
Hi, I'm Julia. I have had the pleasure to be the staff of the Hard Science 360 project, and I just want you all guys to know that I'm very proud of you, and thank you all for your curiosity and your enthusiasm. So please welcome on stage Ina, Mia, Mar, Martina, Grace, Victoria, Wade, Christina, Enoch, Sarim, Guillermo, and Mohammed. Thank you.
Let's start with some adrenaline. So, what would you do if suddenly your heart stopped? Or you have only a few minutes to save a real patient? Believe it or not, this happens every 40 seconds. Good morning, everyone. We're the Hard Science team, and for 11 intense days, we spent time at Belviche Hospital working side by side with professional cardiologists who fight every day against time to save real patients. Today, we bring you the story, the entire story of Montserrat, a patient that changed us forever.
The main question we explored was how cardiologists and their whole team go from the lab to the bench and back in order to provide the best care for their patients.
Wait, 40 seconds.
See, this right here is what happens in the real world. Through many simulations and dummies, we not only learned the processes, but also the importance of each stage in the long journey to recovery. It really helped us in getting to know the perfect treatment for our AI patient, Montserrat. And with these skills, we can even apply them to real-life patients in the future.
As we started with our patient Montserrat, 70 years old, we interviewed her on the specifics of her symptoms, such as shortness of breath and chest pain. However, this was not always very easy to communicate with vulnerable patients, so watch this. How many kids do you have?
I'm not really sure. I don't really count them these days.
After the interview, we were quite suspicious of a myocardial infarction. And this was especially certain when Montserrat called the emergency services in a supermarket. And when the 112 services arrived to the supermarket scene, they performed some tests and recognized that she had gone into cardiac arrest. So they started the process of CPR.
Dala tu cuerpo alegría Macarena, que tu cuerpo es para dar la alegría y cosa buena. Dala tu cuerpo alegría Macarena, Macarena. I'm alive.
So after this, we had to act fast and performing and interpreting the electrocardiogram, which helped us identify that Montserrat had developed an ST elevation myocardial infarction. And the ambulance took her to the cath lab where we treat the blocked artery with PCI and placing a stent and deflating and removing the balloon in order to dilate the artery. However, the next morning, Montserrat deteriorated into cardiogenic shock. The large anterior MI damaged the myocardium, especially the left ventricle, so the heart could not pump effectively. We learned how to assess patients with complications, to decide how to persist, and most of all, keep a positive attitude. We transferred her to the ICU after having observed other patients receiving such close care. We had to operate on Montserrat. Of course, we couldn't do much more than observe the surgeons working, but we mastered our suturing skills. We successfully saved Montserrat and are now tracking her health via telemedical app developed by the research team.
After this interesting case, we worked in the lab to explore the biological reasons behind the patient's deterioration after a myocardial infarction and identifying possible biomarkers through a Western blot that could help predict risk. To aid this, we observed how RStudio and Python can be used as research tools to explore anonymized data sets, visualize patterns, inspect variables, and understand the basic logic of predictive modeling for risk stratification in translational research.
This case taught us that modern cardiovascular care is not built on one skill alone. It requires a medical knowledge to understand the patient, teamwork to work quickly and safely, and creativity to solve the clinical problems to better solution for our patients. From Montserrat's first chest pain to her recovery and digital follow-up, we saw that future of medicine depends on collaboration between clinicians, researchers, data scientists, and innovators.
What makes this project so Montserrat was never just a Montserrat was never just a diagnosis after a diagnosis. She was a patient going through the entire healthcare system. And although this case was a hypothetical one, it is the story of hundreds of thousands of people. And so we are honored to have had the opportunity to acquire so many skills, from puncturing arteries to suturing them, and gain awareness as future doctors. We have all taken our own individual takeaways regarding what we'd like to pursue in the future, but I believe I speak for everyone when I say that these were some of the most reflective and rewarding 11 days we could have asked for.
In conclusion,
We are really grateful for this opportunity. It has marked a turning point in all of our lives, and this project is just a beginning for what awaits us in the future. So we want to give a thanks to Fundació Catalunya La Pedrera, all the staff members, everybody watching us here, our families, every peer we have here. And also a special thanks to Julia, our amazing staff member. You have been great. You made this experience unforgettable, so thank you for that.
During this experience, we also saw pain, urgency, motivation, talent, and hope, loads of hope.
We would also like to thank the medical team who made all of this possible. So, Dr. Comín, no hi ha res més maco que l'ofici exercit des de la vocació, així que per la teva passió, per la teva dedicació i per la disposició del teu equip, gràcies.
And finally, and most importantly, thank you guys for everything. We've truly bonded, and I can't wait to see you again as cardiologists in the future. Okay.
Hi everyone, I'm Caral Nomen, the staff member from Quantum Physics and Cryptography. I just want to say thank you to ICFO and also to my group. Guys, you're brilliant, and also thank you for being such a wonderful people. So please, a big applause for my group. So welcome on stage to Yuri, Boris, Seth, Burak, Yichuan, Pau, Rob, and Octavio.
Welcome. Nowadays, as we've seen in the video, having privacy is almost impossible. Every single day, someone is trying to rob us. It happens to individuals, to banks, to governments. Nobody is safe. Protecting our private information in today's world has become a monumental challenge. Protecting our private information in today's world has become a monumental challenge. From a simple text message to massive amounts of global data, the solution actually lies in the laws of quantum physics. Today, we're excited to show you the science behind how this works using the BB84 protocol, and we will show you how to survive in this digital battle.
That brings us to this question. How does the laws of physics ensure that a secret stays a secret? But first, a little bit of background. In quantum mechanics on the quantum scale, tiny particles such as light photons can exist in an entangled state called superposition. Think of superposition like a coin flipping in the air. As it's spinning, it hasn't exactly decided which side it's going to land on. This entanglement is what makes quantum so unique. This entanglement is what makes quantum so unique, as essentially a particle can exist in two states at once. But the thing is, as soon as you try and measure one of these particles, it instantly collapses into one of the two states. Regarding cryptography, this makes messages almost impossible to decode, as soon as you try and observe it, it'll instantly collapse into one of the two states. These are the forefront of ideas in which our project was surrounded at ICFO. Yeah.
Okay, so the current encryption, the conventional encryption, it is a the problem with current the problem with conventional encryption is that it relies on mathematical algorithms that are expected to be vulnerable to sufficiently powerful quantum computers in the future. So that's why we want to introduce you to quantum cryptography. So how did we manage to simulate the BB84 protocol on a smaller scale in one of the ICFOS labs? Think of a qubit as, go back again. Okay, think of a qubit as a photon, a particle of light that can exist in a superposition state. Now, Alice can sense. No. Go back. Okay. It is sent from Alice to Bob for using a laser, for example. Now, photons can be polarized in one of these four directions: vertical, horizontal, diagonal, anti-diagonal. And so by using a filter, Alice prepares each one of these photons to prepares each of these photons in one of these four polarization states. This way, Alice encodes his message. If Bob happens to measure the encoded photon with the same state as Alice did, Bob will get back the original photon.
But think, what happens if Eve or anyone else tries to intercept the photon? If Eve or anyone else tries to intercept the photon, then by measuring the quantum state of the photon, it might change. So then Eve, in order to imitate the transmission of light and not get caught, she will have to send the information to Bob. But by sending the information to Bob, if the quantum state of the photon has changed, she will get detected.
So at this point, we already knew all the theory necessary to run the simulations, like the practical part, and that's exactly what we did. We simulated a BB84 protocol. First, we had to build a circuit that it was the photon on the right, and that basically was a bunch of lasers aligned that we aligned perfectly using a technique called beam walking. And after a bunch of coding, we were able to run two simulations. In the first one, it was just Alice and Bob, and in the second, we interfered Eve, which tried to capture the message, and both simulations were run successfully.
Above all, the most important takeaway has been learning with extremely valuable researchers. They've shaped us into becoming really good scientists. The way they inspired us into becoming more and more creative, their deep passion for science has formed us into what we have become today, and they inspired us into our future careers and motivated us to push more.
All right, what's the take-home message from all of this? So, all around the world, every day, thousands of people are getting hacked. Thousands of people are having their identities stolen. And this problem will only get worse as the internet and technology become more important in our day-to-day lives. However, there's a solution. quantum cryptography. It doesn't rely on any old methods, it just relies on the laws of physics, and it will undoubtedly be an important technology in the future. Thank you for listening. And now, we will, it's broken. And now we would like to thank Carolta, all the other staff members, and all the researchers and mentors who helped us with this project. Thank you.
All right, hello everyone. We're Julia and Wasima. We're the staff members of the Broadening Medical Expertise at Hospital Clinic. We're super proud of our group members. We hope that you made the most of this life-changing experience and that your past self, the ones who walked in here on the first day, are very proud of who you've become today.
Please welcome to the stage Emma, Paz, Giring, Omar Fadel, Inés, Judith, Lucas, Jane, Armand, Ella, Greta, and Joanna.
First of all, good morning. We're the group of 13 students who for over 2 weeks have been participating in the Broadening Medical Expertise project of BIYSC. During this time, we've worked with numerous specialists in different departments of the Hospital Clinic of Barcelona, including ophthalmology and nephrology. Today, for our final presentation, we will, through a simulated case, present you a snippet of the work through which we've been able to learn from and grow with in these departments. We hope you enjoy.
So today we're going to present a case that it's hypothetical, and we'll walk you through it so you can understand a bit of what we've been learning these past days. So this is a man, this is a 58-year-old male that came to the hospital after after feeling not very well. We'll explain that later. And his background is that he's diabetic for the past 10 years and also has hypertension. So now my colleagues will guide you through this case and what complication this man had because of not following his treatment.
The doctors ordered blood tests because the patient, the doctors ordered blood tests because the patient was feeling very tired, and the results showed that his kidneys were not working properly. They also found high levels of a waste product in his blood, which is a sign of kidney failure.
The doctors also checked his blood sugar and performed a urine test. His blood sugar has been constantly high due to diabetes, and there was a high concentration of protein in his blood. They also measured his kidney function, which was very poor. All these results confirmed diabetic nephropathy.
Because kidneys stopped working and cleaning the blood, the patient started hemodialysis 3 times a week. Dialysis removes waste products and extra fluids from the blood, and the patient will use it until another treatment will be found.
After more medical tests, the doctor decided that the patient was suitable for a kidney transplant. A compatible donor was found and the surgery was successful. After the transplant, he no longer need dialysis.
After the transplant, the patient had to take immunosuppressive medication every day to prevent rejection. He also attended regular checkups to make sure he was healthy.
During an eye examination, the patient said that he had blurry vision and difficulty reading. So the ophthalmologist performed a fundus examination which showed diabetic retinopathy caused by years of poorly controlled diabetes.
During the physical examination, the doctors found swelling in both of the legs, high blood pressure, pale skin, and poor vision. These signs suggested advanced kidney disease and diabetes complications.
Diabetic retinopathy happens when high blood sugar damages the small blood vessels in the retina over many years. If it's not treated, it can lead to serious vision loss or even blindness. That is why regular eye examinations are very important for people with diabetes.
So to treat this diabetic retinopathy, the patient received anti-VEGF injections, which help reduce swelling and slows the damage to the retina.
The patient also had laser photocoagulation, which is a laser eye treatment that seals out blood vessels in the retina, and it also helps protect his vision. Regular eye checkups are also really important to detect any changes early on.
After all the professional treatment, the patient has been cured and regained healthiness. Thanks to the presentation of my colleagues, over the past 7 days in Barcelona Hospital and a research institute, we have experienced the real world of medicine and scientific investigation firsthand. Witnessing the dedication and the professionalism of doctors and the researchers has inspired us to pursue our scientific ambitions and contribute to improve human lives. We are deeply grateful to BIYSC for creating this incredible opportunity for us to learn from leading experts, connect with all talented students from around the world, and building a bridge between present and the future. Thank you, BIYSC, for making this unforgettable journey possible, and appreciate our staff member, Julia and Wasima, their contribution for us in the past few days. And these are our memorable moments throughout the program. And thank you all for listening. Thank you.
Good morning, everyone. My name is Neil Salat Rovira, and I was the staff member designated for the Neurobiology of Trauma project, Effects of Behavior and Brain Function at the Institute of Neuroscience back at the Universitat Autònoma de Barcelona. It's been an absolute 2 weeks of fun, pizza, sleeping pictures, and all of that. So please welcome our trauma gang, Harry, Celine, Alicia, Laia, Mathilde, Alejandra, Anastasia, Naomi, Laia, Sudan, and May to the stage. Thank you.
Hello everyone, we are here to present the Neurobiology of Trauma, Effects on Behavior and Brain Function.
Okay, I want to start explaining the background of the project. And 70% of the adults have experienced a traumatic event at least once in their lives. Moreover, depressive and anxiety-related disorders are more prevalent in females than in males. And finally, our aim was to investigate the relationship between sex and stress using rats and mouse.
So, the aim of this project was to answer the question, which neurobiological factors are making females more susceptible to the effects of trauma? And why are we focusing on females only? Do we hate men, maybe? I don't know. But the real reason is, in the past, we have been focusing more on male studies because females have pregnancy, their hormones really are altered monthly by great degrees. And so it really complexified the studies. So by studying the effects on women, we are capable of providing treatments synchronized according to their menstrual cycle, because we know estradiol, for example, helps the recovery of trauma greatly. Also, we can improve other aspects of medicine that are also affected by gender. For if hormones are affected, then definitely a drug will act differently on a woman compared to a man.
Okay, so first of all, what is PTSD? PTSD is a condition characterized by the appearance of symptoms after an event that is stressful, extremely traumatic, or involves physical harm. In the case of this project, a trauma response was induced in mice by immobilizing them for 2 hours in the position seen in that image. This obviously brings up clear ethical concerns. However, researchers combat this by providing the mice with the 5 freedoms and the 3 Rs. Additionally, us as students did not have direct contact with the mice and the rodents themselves, and we used previously obtained videos and results from other studies done in the past.
During the project, we did genetic analysis. Our goal was to determine which of the mice had a genetic mutation of CRE. CRE is a gene, or it's a site-specific recognition technology that reacts with a genetic sequence, like the one you see in the corner, that is twisted and untwisted and able for the protein to be synthesized. This does not occur in the wild, so it's very important to find which mice had the gene. So we injected the mice with a CRE-dependent dread virus, and like you see in the corner, we were able to see which ones reacted with the CRE.
Here we have 3 possible combinations of these alleles. We have 2 homogeneous and 1 heterogeneous. There are the Y tip and the mutation, and also the Y tip and the mutation that are indicated in the tap lines.
We also studied the estrous cycle, which is really important to understand the female brain work due to the hormones affecting the brain. The estrous cycle contains 4 phases, which is proestrus, estrus, metastrus, and diestrus.
So as you can see in that middle image, the estrous cycle has 3 different types of cells that are present in our samples, which are those. And the 3 types are cornified, leukocytes, and nucleated cells. So depending on which type of cells are in the samples, we can tell which phase the mouse is in. And depending on the phase, there's different levels of a hormone called estradiol, which can affect the extinction of fear memory. So if there's more estradiol, then the mouse experiences less freezing due to their fear memory compared to if there's less.
Okay, so we did some series of experiments at the start of the course talking about what the effect trauma has on the behavior of the animal maybe 3 or 4 weeks after the event has happened. So we use these 4 tests, which you can see here. The elevated maze test on the top left. On the bottom left, there is a social interaction test. On the top right, there is an object interaction test. And the final, on the bottom right, there is a forced swimming test. So what we examined is we compared a sample of rats which have experienced trauma through immobilization. And with them, we had those which haven't experienced trauma, and we analyzed the behavior in these tests. And we saw a marked difference in how they behaved in these different tests. For example, in the elevated maze test, as you can see, it's a maze shaped like a cross, but it's elevated maybe 40 or 50 centimeters above the ground and has half of it exposed. Obviously, there's an instinct of the rats to explore, but those who had experienced trauma before spent much less time in the open sections and preferred to stay hidden away in the closed sections. So we had a lot of data like this, and we processed it with all of these tests, which indicated that trauma really does affect the behavior, even a smaller model of an animal like a mouse or a rat, which was interesting to see, especially as we processed the data ourselves.
Immunofluorescence microscopy is a microscopy technique which employs the use of fluorophores to bind to proteins of interest.
to help enhance visibility under the light of a epifluorescence microscope. After exposing the rodent to a traumatic experience, like we previously discussed, the brain is then extracted from the rat itself and preserved. After unfreezing the brain and making it undergo a incubation process, the concentrated light from the microscope will be able to shine onto the fluorophores, exciting them, which will then refract more light into the microscope for you to see the images in a variety of different colors depending on the light which you project onto it. In the left side of in your right of your right side of the screen, you'll see the samples that we went through. The two brain fragments are like the same brain, but just of course under different lighting. And then on the very top right, you'll see a enhanced version of that thing. The little white dots that you can see are stress hormones known as CFO. In rats that had experienced the trauma, the CFO were much more concentrated and dense, whereas those who hadn't had a lot less dose of CFO.
These are just some throughout the experience, and then okay, so our final words is okay, so after this experience, one thing that in neuroscience is not typical is lab work. It's very hard to find lab work, especially at our age. Our age, and then it was such a grateful experience, and we're very glad. And now that we know kind of the basics of neuroscience, especially how it links to psychology with PTSD, and we're very grateful. And I just want to say a big thank you to Neil, who was able to make this experience just much more enjoyable and always there to support us, and especially all of our researchers and Lydia and Mariana, who are our main coordinators and who are always there answering our questions. So thank you.
Hello, I'm Bru. I'm the staff who has been accompanying this amazing group of people who have been working with infectious disease at Hospital del Mar, and they've been doing an amazing job learning on HIV, STIs, and especially what it takes to be a doctor. So please give a big round of applause to Isabela, Matilde, Ignacia, Jena, Zinin, Miguel, Bastian, Mateo, Annika, Zijin, Anael, Ainhoa.
During the last few days, we have been learning about the art and science of medicine and infectious diseases at Hospital del Mar. Of all the things we learned, we are especially interested in understanding how effective doctor-patient communication shapes the prevention, diagnosis, treatment, and of sexually transmitted diseases.
Doctor, come quickly. I think I'm going to die. I'm a strong, sporty man, but recently I've been feeling so tired.
A 35-year-old male presents to the ER with severe symptoms.
Good afternoon. I'm Dr. Ainhoa, and I'll be consulting you today. This is my resident, Dr. Bastien, and we're going to proceed with a medical interview. So first, we already know your basic information, so we'll proceed with your symptoms, then your medical history.
Yeah, blah, blah, blah. Okay, so let's go to the important thing. Have you been sexually active recently?
What? Oh, of course not. At least not in the last 5 years, maybe 4 or even 2, I think.
No, that is not how you do a medical interview. That was the worst interview I ever seen. You have to be respectful and also a structural matter, and you also have to build trust with the patient so the medical interview can proceed just fine. Go back. I'm so sorry about that. As you can see, he is new.
Well, I'm sorry, sir. Let's try again. So, what symptoms are you feeling right now?
Well, I've been having a low fever for around 3 weeks, and I have this tender bump on my throat. I don't know what's going on.
So, are you on any medication, chronic diseases, something like that?
Nope.
Yeah, sure. Okay, any more symptoms that we should know?
Well, I've been experiencing night sweats recently. Night sweats. I don't know. I think it's because of the fever.
Fever, fatigue, a swollen single note. Dr. Bastian, what do you think this patient has?
I think we need some tests.
A few days later.
Okay, so the test arrives and it's not good news.
Sir, we are so sorry to inform you that the test has come with some unexpected result. The test of HIV has come back positive, but we want to let you know that we are here to support you, and we will also additionally have to test on your sexual partners to prevent more infections.
Freeze. Let's pause this medical drama to explain what HIV actually does. Here's how it works. HIV finds a healthy immune cell and forces its way inside. The virus attacks the cell, forcing it to make thousands of copies of HIV. The new viruses burst out of the cell, killing it off and go off to attack more cells. Because the virus is spreading so quickly, his immune system is fighting back hard. That battle is exactly what causes his fever, swollen glands, and night sweats.
Doctor, what can I do now? Can't I take PrEP or PEP?
I'm so sorry, it's too late.
PrEP and PEP are prevention treatments. PrEP is a resource for people who are at risk of getting HIV, and PEP is a short dose of antiretrovirals that prevents an infection in the first 72 hours after an accidental contact with the virus.
So now I just wait to die?
No, of course not. Although HIV is not curable yet, we can prescribe you some art to fight the infection.
Art? I'm not Picasso.
Art, apart from the art, means also antiretroviral treatment. It's basically a pill you take or an injection that makes the virus stop replicating or slowing the replication process. So basically, it saves you from dying. As you can see, here's never mind, okay.
Okay, so if I take art, I will still be fine, right? I can live a long, healthy, normal life?
Well, of course you can. I'm sure you will have a long life and happy life. Of course, if you stop taking the medication, the virus will start replicating, and you probably, if you have a irregular consumption, you'll get AIDS, and that's very bad.
Okay, thank you so much for your nice words of support. I feel so much better.
So, what have we learned today? You must never judge a patient. Their trust is the base of a successful patient-doctor relationship. Also, in medicine, asking the right question is just as important as giving the right medication. The 4 bioethical principles are the compass of medicine: autonomy, beneficence, non-maleficence, and justice. STIs are also preventable and can affect everyone. And they do face a great amount of stigma. This is why you have to remember that it can affect everyone and anyone. And education is the key to overcome this stigma. STIs are also treatable. HIV can be treated with ART, or ART as they said, which lowers the viral load. And also, you always have to remember U equals U, undetectable equals untransmissible.
Thank you. Lastly, we wanted to say a special thank you to the Hospital del Mar, Dr. Roberto Guerri and all his colleagues, and to Brew for making this amazing 2 weeks possible. Thank you so much.
Hello everyone, I'm Ines, and I've had the pleasure to be the staff member of the molecular breeding team, which is also known as the melon team. So, to be honest, I don't have enough words to say how proud of them I am, and I have seen how they have made an amazing team at the lab, but further than that, I've seen how they've become friends, and I'm pretty sure they're gonna stay friends forever, so please welcome to the stage to Giovanna, Sofia, Nora, Agatha, Elias, Arde, Panchika, and Amber. Also, special mention to Irene and Ardin, who cannot make it because they left yesterday, but they're also a big part of the team.
All right, imagine you're walking on a melon farm on a warm afternoon. Some melons are very small and dense, while others are very large. Some taste very sweet and some are very firm. It's like each and every single melon has its own characteristics. So, what shapes a melon? Well, what decides its traits? Let's try and unveil this mystery with molecular breeding and gene editing to feed the world's population.
Due to climate change and the growing population, we have a necessity to increase the yield as well as the nutrition of our crops. In our case, we designed a melon that has orange flesh, which is filled with beta-carotenes. And beta-carotenes are naturally converted to vitamin A. Vitamin A is an essential vitamin that plays a role in immunity, growth, and eyesight. Therefore, we crossed a white flesh Pio de Sapo, which has an extended shelf life, with an orange flesh Vedrante, to get an ideal melon, as you can see here, which is both long-lasting and nutritious.
So some of the potential solutions of the problems that we're facing nowadays are things like gene editing and molecular breeding. So for gene editing, right now one of the preferred methods is CRISPR. CRISPR because it's very simple to operate, and also because it gives us the chance to select a specific section of the genome, so we can identify it, we can find it, and then CRISPR comes in and snips it sort of like a pair of scissors. So after this, we can introduce a random mutation into the gene, or we can even insert a specific engineered cut of DNA. As for molecular breeding, that's more like a modern way of artificial selection. So when we find a specific trait that we like or that we're interested in, we can find markers in the DNA to help us know exactly in which position of the molecule it is. So that way, with specific types of breeding, we can ensure that this gene stays within the next future generation so that we can still keep this characteristic in our future plants.
So the first step in finding the gene behind the color orange is by crossing two types of melon. One, Vedrante, and the other, Pio de Sapo.
Once the parental lines have been crossed, an offspring is created. These offspring can be analyzed in a lab to see traits. The ones that we got are from Torre Marimon, which is the greenhouse from the Crag Research Center. We got the melons back to the lab, and there we began the phenotyping process. This means analyzing characteristics or seeing how genes are expressed.
So in order to phenotype each melon, we first measured each one's weight, then we cut them in half and we assessed the rind and flesh color. Then we used different tools to find the firmness and the sugar concentration, also known as the sweetness, and then we scanned each one to find the height and weight, or the height and width. This was all really important qualitative and quantitative data that we use later.
To identify the genes responsible for the traits that we were studying, in this case the color of the melon, we had to extract the organism's DNA, so the melon's DNA. To do this, we first collected sample tissues from the leaf of the melon plant and broke its cells to expose the DNA. However, because now our sample consisted of proteins and sugars in addition to DNA, we had to purify it. To do this, we used a chloroform and centrifuge machine to isolate DNA. Afterwards, we had to assess the quality and quantity of the DNA that we had extracted. To do this, we used the gel electrophoresis and spectrophotometer to see if it was an adequate sample. When we determined if it's adequate, we were ready for genotyping.
Genotyping is a technique used to identify specific genetic differences.
We did this and converted our data into a genetic map, as shown in the middle, which is a map of the order and the distances of the markers in the DNA. We then combined this data with phenotyping data to create a chart showing the likelihood of a certain trait being located, the likelihood of a certain trait being related to a certain piece of DNA. This gave us a rough idea of where in the DNA our wanted gene was located. And then to get more specific, we can always just cross these plans, this data with data from other genetic maps, and thereby make it more narrow.
To even narrow the area as much as possible, we plugged it into a J browser, which is a website where you can find the whole genome of a melon. And there we got 11 candidate genes. And after analyzing each of their function, we got our specific gene called protein orange, which is the gene we wanted.
Finally, we proved that we had selected the correct color-determining gene by using the CRISPR method mentioned earlier. And we did this in order to try to silence the gene that makes the melon orange. So in order to do this, we took orange melon seeds, like the one from this, and we prepared them, and we infected the melon with an agrobacterium that is able to alter the DNA of the melon. And so once they grew, we could cut them open, and if the flesh is white, then we know that we've selected the correct gene, and we can use that information in the future to create more orange melons.
So what's the take-home message for this project? Well, throughout these weeks, we've worked with melons, but this project is way, way bigger. So as the population grows, and with climate change and limited resources negatively impacting agriculture, we need more innovative solutions. And that's exactly what we can use genomics to solve, where we can identify specific genes behind its traits, and then use molecular breeding and CRISPR in order to make improved crop varieties that are more resilient, more nutritious, and are more sustainable. So the whole point of this is actually looking at our crop at a molecular level in order to actually identify the gene, because the smallest change in a single gene could make the biggest difference for millions of people.
As a final note, we as a group would really like to give our thanks not only to the foundation, but also to the people at CRAC, to Sylvia, to Marta, to Montserrat, to Alejandro, and to Sergio. Only the entire presentation. And we'd like to thank Ines specifically. She really helped us connect a lot more. And we would like to thank the foundation and BIYSC as well for bringing us all together and letting us have these unforgettable experiences. We really have here, not only within our group, but also with the other people in the program. So thank you all for being here as well.
Good morning, I'm Mireia Rizo, the staff member responsible for the Advanced Mathematics group. It's been a pleasure to meet you guys and to spend these last 2 weeks with you. You worked really hard, so I'm very proud of you. So please welcome to the stage Lucia, Rawaf, Arjun, Lohit, Matthew, Santiago, Sofia, Leire, and Pavel.
Good morning. Thank you all for being here. We are the Advanced Mathematics group, and during the past 2 weeks, we've been taking part in 4 different types of lessons to study how our mathematical theories applied in the real world.
So our first lesson included maglev trains. These are magnetic levitation trains, which are magnetically propelled across the track. So this means they can go extremely fast. But how do they slow down? So they produce a repulsive magnetic force by reversing the polarization of the magnets. So this means that they can decelerate at rates beyond 10 meters per second squared. Well, this is extremely dangerous. So they need to decelerate at 1 to 2 meters per second squared. But this is easier said than done, because how are the engineers and track companies supposed to know how long to make the track?
So this is the model of the train, and this is rubbish. This leads to this equation. Now it looks quite simple, but it's actually very complicated for a couple of reasons. Firstly, there's a lot of units, so all of the B, C, A, M, V, they all have different units, and that makes solving this very difficult. But we can actually, as you see on the right, we can kind of help to simplify that and do some, and sort of start to solve that. The second thing you need to take is that the C is very small. So we can use what's called a perturbation to help simplify the problem and then introduce it later. So what we're going to do is we're going to take some stuff out and then put it back in. So, this is rubbish. So, you're not expected to follow along with all of this, but basically it's just to see like what is going on. And now I'm not even pressing the button and it's just going. Okay, well, you know what? You can see what's happened. First of all, we took out the C, we set it equal to 0, we took out all the units, and then we simplified it. It's still going, I don't know why. And then we got an equation that we had to substitute the numbers in, and we get a solution, and it's going to come up in like a minute. There, the equation is coming up. It's like, whatever. Like, you don't need to understand this. It's a waste of time. But the point is that you need to find the time and you need to find the length of the track. And with a lot of computation, eventually you get some functions that you can solve. Oh, okay, wait, we're only halfway. So you need to find a speed of the train where it's like almost stopped. So then you plug in all the numbers again. Wait, the numbers are coming in. Yeah, there you go. That's the first time. We can then slightly improve that a little bit. It's nearly done. I promise I'm going to stop talking in a minute. These are the equations. There you go. That's how long you need the track to be. You need it to be 6,900 meters, or all of your passengers will crash.
Markov chains. Markov chains are a mathematical way to describe a sequence of events. These are characterized by the Markov property, and where this feature affirms that the next state depends only on the current state. So the present has all the information we'll need to predict the future. Markov chains help us understand situations where outcomes are uncertain but follow specific patterns over time.
All right, so as you can see here, there are some real-world applications of Markov chains. We've got random sampling. Go back a bit, please. Yeah, random sampling, navigation, machine learning, and weather forecasting. Now, as you can see here, there's a cheeky diagram predicting the weather. So let's say it was sunny for one day and the next day was sunny as well. You have an 80% chance of that happening. You can see from the arrow going out the sun and go back into the sunny. Now, obviously, that's way too simple. So next slide. Yep, that's the one. We got a diagram here. This is a simulation that shows all the possible random walks as you repeat them over and over again. Now, obviously, this looks very complicated, but the average of this is actually quite stable. But then as you do more, the possible outcomes increases.
So for our third project, we did fluid trajectories. Well, we can't study a fluid by its entirety, so we do it like particularly. So we basically, by the Euler method, which uses a particle current velocity to calculate its next position, And by doing this over some time, we actually get an approximation of the trajectory of the fluid, and that's the equation at the right down. Yn plus 1 is the next position, Yn is the current position, h is the small time steps which the function is measured in, and f is the position of the x-axis and the y-axis. But this method doesn't consider neither density, neither the length of the trajectory, but we can use like the density or the viscosity actually to by using the Navier-Stokes equation, but that's too difficult. So we only calculate the length of the trajectory by using the trapezoidal rule, which basically is an integral, but it's approximation of the length of the trajectory by using like trapezoids to calculate the area below the curve, like that.
Well, and basically, after applying maps, you will get this diagram of ocean. You can do the same for atmosphere as well. And this can be applied to designing airplane wings, so like they don't break in the air. It can be also applied to weather forecast and building offshore wind farms.
And lastly, we'll be talking about discrete iterations and dynamical systems. So dynamical systems are defined as a change in a system state over time according to a fixed rule. Particularly in logistic systems, we are able to describe and predict the population behavior of a species.
Yeah, so in here we have the logistic equation. It describes how a population changes, like from one to the next one. And as we can see, the x is the population, like the growing population, and r is the growth rate. And over here, we have the logistic curve that, as we can see, at the start, the population grows rapidly, but then it just slows down, and then finally it stabilizes.
No, go back. We create models by first simplifying them through assumptions and then later on describing the movement using functions. Particularly for this simplified model, we can later adjust it depending on the situation we're applying it in.
Yeah, so meaning that a good model is a good that is going to be effective and is going to be useful for a particular time and situation.
And lastly, real-world application of logistic models is, for example, help predict bacterial reproduction rate, which then helps biologists model disease outbreaks.
Yeah, and also, so I'm going to explain what a logistic model is. So a logistic model, it's a nonlinear mathematical model that, unlike exponential growth, it takes into account like a mortality rate. So population cannot grow infinitely.
Right, to wrap things up, maths is used everywhere in the real world and in the quantum world. I'd like to thank the researchers for everything they've put into it. I'd like to thank the BIYSC staff, and lastly, special thanks to Maria for putting up with us for the last 2 weeks. Cheers.
Hello everyone, my name is Joan Zonneberg, and I'm the staff member for the Supercomputing for Next Generation City Design, which took place at the Barcelona Supercomputing Center. I just want to say to my kids, I'm so proud of you, and I want to thank you for the laughs, for not getting lost on the metro, and for amazing memories that we're all taking home with us. Please welcome to the stage Ian, Aritz, Haven, Rayana, Abdul Rashid, Mia, Rand, Madvi, Heya, and Ahmed.
Okay, so here we have a diagram that shows the populations of both Riyadh and Barcelona. And as you can see, the population of Riyadh is much, much bigger. So I want to ask you guys a question because I can see some of you falling asleep. First of all, I want to ask you, do you think that Riyadh will have a higher number of public transportation users than Barcelona? If you think Riyadh will have a higher number, raise your hand. Ooh, only a few. And if you think Barcelona, raise your hand. Okay, I can see why you guys got accepted into BIYSC, because the answer is correct, it's Barcelona, in fact, and the difference is actually huge. And as you can see, there are a lot more public transportation users in Barcelona than Riyadh.
This leads us to our research question. Why does Riyadh have a higher population but less public transport users than Barcelona? So here we have both images of both cities. The answer could be walkability. On the right, we have Barcelona, which has sidewalks, and it creates interaction with pedestrians. There's trees for shading, and there's tactile flooring for the visually impaired. And then on the left, there is Riyadh, which has little to no sidewalks, which shows that the city was built for cars instead of pedestrians.
Okay, now, to confirm this hypothesis we had, we needed to perform some spatial data analysis with open source programs. So this is what we got. We fetched from these sources some street graphs and the location of some essential amenities we decided. And we all got this from OpenStreetMap. It's a free website that you can search. It has a one-to-one representation of the world, and it is free, public, and you can collaborate on.
Yeah, so in order to compare the two cities, we used rose diagrams, which are these little circles down here, and they are the representation of the street's average street orientation. So if we compare the two layouts, we can see that Riyadh has a more dispersed layout, gridded along motorways, and also has a more segregated land use, which contributes to lower population density. If we look to Barcelona's layout, we can see it is a more compact gridded layout that has mixed-use blocks and maximizes land use efficiency, and also contributes to highly population density.
So to add off of that, we actually generated these maps, which basically show the citizens in which can walk to certain amenities. And this is all within a 15-minute radius. We actually built basically a web application with a front-end kind of portfolio that was able to not only calculate these kind of radiuses, radii between points, and it was also able to store and visually show our data on this map.
So from Haven's data that we saw before, there are 10 times more amenities, so essential services like shops and supermarkets per square kilometer in Barcelona compared to Riyadh. And that just shows that Barcelona is a much more urbanly dense city that basically allows for services and access to services to be much easier.
So we did a demo calculation for a fragment of both cities. In Riyadh, only 10% of people live within a 15-minute walking distance for daily essentials. And in Barcelona, that's 74%.
In terms of accessibility, we found that in Eixample, almost all amenities are reachable within a 15-minute walk, whereas for Riyadh, it's better off taking a car.
Walkability plays a key role in our ability to access essential services and should be treated as a requirement rather than a privilege. Our proposed improvements include planting more trees for shade and cooling, bringing amenities closer to homes, and redesigning streets by building wider sidewalks and adding crosswalks and disability ramps.
All of this was made with open data provided by ordinary people just like you and me. We can be part of the research projects by contributing to open source projects like OpenStreetMaps and raising awareness of importance of making our cities people first and accessible. Stop designing for cars, start designing for people. And we would like to thank all BIYSC staff and our researchers that helped us to create this presentation and helped with tools and visuals. So thank you for all of you to give us an opportunity to participate in this project and make good things.
Hello again. I'm Nuria, and it has been a pleasure of being the staff member of the Innovation in Photochemistry project. You guys did such a wonderful job. I'm really proud of you. I really enjoyed when you sang on the bus and also in the lab. Those days were great. And I'm sure that you will become a great scientist one day. So welcome on stage, Yicheng, Lian, Barbara, Jana, Max, Yaprak, Ilya, Nika, Susie. Okay.
Today, we'll be introducing innovation in photochemistry and drug synthesis, shaping a sustainable future. During the past 2 weeks, we worked on tackling these two main questions. How can photochemistry be used to produce more sustainable energy? And how can synthetic chemistry be used to improve global health? But first, let's look into how photochemistry works. As you can see here, the atoms are in their ground state. But when a photon transfers energy to the electron, it brings it into the excited state. During its excited state, the electron will emit a photon and later return into its ground state. This process is called fluorescence. This can be seen in various places, such as the ones on the screen.
The world has a high energy demand, but the main struggle is to produce it sustainably. A promising solution is green hydrogen production from water. Here we can see some water molecules floating around. Our goal is to split the H2O into diatomic molecules of hydrogen and oxygen. Because H2O is an extremely stable compound, as seen by its abundance on Earth, it needs energy and a catalyst to split it. In this case, the energy is provided by the photons from the sun, and the acting catalyst is titanium dioxide attached with platinum. As the production of this reaction, we get 2 moles of hydrogen and 1 mole of oxygen gas. Then the bonded hydrogen can be stored and later used as fuel for energy.
Another key aspect of our program was to do with solar cells. Solar cells harness the photovoltaic effect to absorb energy from the sun and change it into electricity. In our case, we learned about dye-sensitized solar cells, or Gratzel cells, which use a dye as the material that absorbs the photons, the photosensitizer, which then releases electrons and allows them to pass through the semiconductor and into the circuit to produce a flow of electrons, or in other words, electricity. And now we need your help. Can everyone in the audience shine your mobile phone flashlights to the stage, please? Also, did you guys know, if we capture all the solar radiation for 1 hour only, we can obtain 173,000 kilowatts of electricity, and that can power 1 year of the world's whole energy need.
For our second research question, we ask ourselves, how can synthetic chemistry be used to improve global health? Since ancient history, humanity has fought off disease using chemicals capable of altering our own biological activity. Known today as drugs, these chemicals often have advanced molecular structures which require extensive processes to assemble. Drug molecules are given their unique properties by atoms or groups of atoms called functional groups. These functional groups can be added to molecules or modified in chemical reactions. The sequence of chemical reactions required to synthesize our desired drug from the starting molecule is known as a synthetic route. During the BIYSC, we were tasked with producing a common drug, paracetamol, from 2 chemicals, acetic anhydride as well as 4-aminophenol. This reaction was preceded by a nucleophilic addition elimination mechanism, gave us great insight into the world of drug synthesis. This is because once we had synthesized the paracetamol, we had to carry out a purification process to ensure the quality of our product. I have the paracetamol right here. This is something we made.
So, to summarize, there are many different ways to harness the energy from the sun into more sustainable types of fuels. Green hydrogen is an extremely promising fuel for its non-harmful waste products.
Honorable mentions that we didn't include in our presentations. We oxidize methane using plasma. We visited and learned about how X-ray scanners and electron transmission microscopes work and explored the process of artificial photosynthesis.
We are extremely honored to work alongside several distinguished postdoc researchers from ICIQ for 2 weeks. Also, we as a group want to say thanks to our supervisor, Nuria, who walked us through this exciting journey.
Hi, my name is Paul. I'm the staff member of the Acoustic Engineering group. Yeah, I see you, Samir. So, in the past 2 weeks, we've been able to discover La Pedrera far beyond where tourists go as acoustic engineers. And I'm really proud of your work and of the final result, and thanks to the researchers for your hard work. So please welcome to stage Rebecca, Noah, Feiruz, Sara, Laia, Abdul Mohsen, Yusuf, Yazan, Samir, and Nils. Here's the pointer.
Hello, everyone. So we have been studying at La Pedrera, and we have been trying to answer this question. Back one. Okay. So many tourists visit La Pedrera when they come to visit Barcelona. It's one of the most beautiful sites in the city, and it's really worth a visit. But oftentimes when they're walking along and looking at the beautiful architecture of the building, they'll have their little ear audio guides in, as demonstrated by this with their headphones, and they'll be missing out on one important aspect of it, the acoustics. Acoustics is a really key part of architecture, and particularly what's so fascinating about Gaudi's architecture, because he really commanded sound in a way like no other architects did. And acoustics is a really important part of architecture. take this room, for example. It's a very large room, yet you can hear me perfectly fine. And that's not just because of these speakers. The design of the roof and the curtains are clearly maybe they need to work a little harder. But the curtains and the seats and the design of the roof are all contributing to you being able to hear me well. And acoustics are also important for simple roofs that we live in, because it impacts our psychological mind as well. So now we will explain a little bit about what we did.
So before my colleagues start explaining the process of what we did, I'm going to give a very brief summary. So the first thing we did was go to La Pedrera to basically get the parameters of the rooms we chose. We popped balloons to get the response of the room. Then we went to La Salle, where we went to the anechoic chamber, recorded the pure sound without any reverberation of different songs we wanted, and then putting those together through a process called auralization, we basically, with that one recording, we made it sound as if we were playing in those rooms at La Pedrera, and you will see right.
Okay, thank you, Rebecca. So we're coming into the process. So we've gone to lab work and also immersive work through multiple stuff, taking 360 images at La Pedrera. The reason why we're doing this, so we're able to create an immersive experience. Not only that, we were able to pop balloons to really get all the frequencies from La Pedrera. We were able also to scare a lot of the visitors there. Next slide. Okay, so in anechoic chamber, we'll able to record sounds. We also play the popular song, very popular song, We Will Rock You. It's a very beautiful song, and it's a very perfect example for the situation. As you see, we're really able to enjoy our time here. So after we record those sounds, we're able to go to post-production studio and really see on those sounds.
So, after going to Casa Milà twice to take measurements and to record audios, we started preparing the VR experience. So what we did, first of all, we 3D designed some of the rooms so that we could calculate their volume and later use this for scientific research we will show you at the end of this presentation. After that, we processed audios using a software called Audacity, which we use to cut them and get them ready for simulating how it sounds at La Pedrera. Okay, so, nice. After we simulated the sounds would sound at La Pedrera, we put it all in a video, in a 360 video, so that we could wear VR headsets and look at it and hear it as if we were in La Pedrera. So we had a fully immersive experience. And
Okay, so now we will see our video where we will show you a song that we recorded in the anechoic room at La Salle, how it changed because of the acoustics in each room of La Pedrera, and because of the type of architecture that Pedrera has.
Face in the sand when I picked it up, then it vanished away from my hands. Down, I had a dream I was 7.
To come to the conclusion of our project, La Pedrera Not only architecturally is it special from other average buildings, but also acoustically. And why? So we gathered some data, as you can see on the graph. We have reverberation time of different rooms. And in La Pedrera, we can see that it is lower than an average building, the red part. And lower reverberation time means that it's more comfortable to our ears. And this tells us that La Pedrera is comfortable for our ears and makes it acoustically special.
So what we take home is that Gaudí combined architecture and acoustics to create a unique experience. Thank you, BIYSC, for the opportunity. Thank you, Paul, for supporting us, and thank you for all that participate to make this a unique and unforgettable experience.
Okay, hello everyone. I know you're tired. This is the last presentation, I can assure you. I'm Sarah. I'm the staff member of the Josep Carreras group in the Multi-omics against leukemia. And I just wanted to say that I'm so proud of you guys. You have been doing a really good job in these 2 weeks. And I hope you have learned a lot from the researchers. And I'm gonna miss so much the party bus with you. So please welcome to the stage with a big round of applause to Matilda, Bianca, Caral, Anne, Sinoilin, Adriana, Aurelia, Nada, Didac, Vero, and Carmela. You guys, you have this here.
Good morning, everyone. Today I'll be introducing our research question, which is, how is the P10 tumor suppressor gene altered in T-cell acute lymphoblastic leukemia patients regarding genomics, transcriptomics, and epigenomics, and how does that affect their response to therapy?
Okay, so first, leukemia starts in the bone marrow, and it starts as a stem cell. Then it can differentiate into all types of cells. In this case, they can divide into myeloid cells or lymphoblastic cells, which are the ones that we focused on.
T-cell acute lymphoblastic leukemia is far more common in children than in adults. However, we used adult samples in our project because they are the ones that have a harder time recovering from this disease.
Some of you may if genetic diagnosis and testing is actually important, and the truth is that it really is. In this image, we can see that in the beginning, we have some cells with mutations, which makes the cells resistant to chemotherapy. Before chemotherapy, these cells are just a minority, but after being treated or after chemotherapy, most cells die except for these resistant ones. So they survive, proliferate, and end up being the dominating population. With genetic testing, we can identify this and develop new targeted therapies, which are currently being used to treat patients alongside.
P10 is a tumor suppressor, which means it tells cells when to stop dividing. If it doesn't work, cells will proliferate without going through checkpoints properly and will become tumor cells. Each omics level studies a different aspect of P10. Genomics studies the mutated one, epigenomics methylated, and transcriptomics gene expression.
Okay, so the first level of multi-omics is genomics, and first we extracted DNA from the patient's blood, and then we amplified the DNA fragments that contain the P10. And then we performed next generation sequencing to identify if there were any mutations.
So within epigenomics, we studied DNA methylation, which is basically the addition of a methyl group to a cytosine that's followed by a guanine, but without changing the base sequence. So basically, this addition makes blocks basically the promoter region of the gene, which is the beginning of the gene, and it basically prevents the PTEN gene from being expressed. And yes, and this is very important because because the inexpression of the PTEN gene can be one of the leading factors affecting leading to the development of this disease.
For the transcriptomics section of the multi-omics approach, we extracted the RNA from the DNA samples and then we isolated it, and then we did reverse transcription to convert it back to cDNA. And we also used the PCR method to target and amplify the PTEN gene because that's what we're focusing on. And then through gel electrophoresis, we were able to see whether the PTEN gene was expressed or not for each one of the patients.
So here basically we have two different cases of our patients, like dividing it in the different multi-omics that we've done. So within the gene, we can have the expression of the gene or not the expression of the gene. So if the gene is expressed, it will have the protein. Well, it might have the protein, but if the gene is not expressed, it means that more or less it's like silenced, so it may not have the protein. Then we know that within the promoter of the gene, we can have it hypermethylated or hypomethylated. If it's hypermethylated, it will probably affect the gene expression and not express the gene. At last, within the sequence of the DNA, we can have it mutated or not mutated. The one that is mutated will affect the protein and make it non-functional or even have no protein. And if you have it a non-mutated, you will have probably a functional protein. So here we have two different cases that we got.
Adding to the slide, the PTEN agonist is a molecule that mimics the function of the PTEN gene. So when a patient is given the drug, they are either respondent or non-respondent. Right. So if the gene is not expressed, then that means the drug acts as a tumor suppressor. If the gene is expressed, the drug doesn't work well and cannot actually work as a tumor suppressor because the gene is already there.
Right, so what is our take-it-home message? Leukemia affects around 2 million people across the globe, and in the future, we will need even more research on tumors and similar diseases, as well as more innovation in technology and other research fields. So our goal should be to find a cure that is targeted, embrace research, and cooperate with various research fields, just like we do at BIYSC. If we all contribute, just like the tens of thousands of researchers across the globe, we can save those affected by leukemia or other similar diseases together.
With that being said, we wanted to thank the researchers from Josep Carreras Institute, Eloy Santos and Ainhoa Perez, who isn't here sadly, and also for being such great teachers and for making this such an enriching experience for us. And last but not least, also to Sarah, our staff member, for being the best.
We also want to thank the Fundació Catalunya La Pedrera and the Josep Carreras Leukemia Research Institute for providing us with this incredible experience. And I also want to thank all of my teammates because they have done this experience unbelievable and unforgettable. And lastly, we want to make a little shout out to Eloy back there with one of his amazing quotes, because it's good to be one, 1, 2, 3, BIYSC!
Thank you all. Guys, what an incredible set of presentations, for real. I think that everyone in this room will agree with us. We're super proud of everything you have achieved, and you should be too. Now we want to express our gratitude to the research groups and research centers collaborating with BIYSC. Thank you for opening your doors, for sharing your knowledge, and for inspiring all these talented students. Your collaboration and commitment and enthusiasm is essential for making BIYSC possible, and it's an invaluable collaboration for the science programs at Fundació Catalunya La Pedrera. And of course, a special thanks goes to the education and science team at the foundation. Thank you, Monica. Thank you, Eva, Gerard, Berta, Nando, Pau, Luis, Marin, the communications team, and the rest of the team for your hard work, for your support. It's a pleasure working with all of you to nurture this talent and for making all of this possible.
We would also like to thank the families for your support and for believing in your kids. Thank you for being here, the ones that are here and the ones that will listen to this afterwards. It's a pleasure, and thank you for trusting us with your kids for these 2 weeks. A big acknowledgement also for the staff team. It has been a pleasure this year to have you here. Your passion, your kindness, your support has been amazing. Thank you for being here and making this 10th edition the biggest one and the most successful one. And then last but not least, to you participants, thank you very much for being here, for your curiosity, your passion, and your teamwork. We hope you have had the best experience possible. It has been amazing to see you grow during these 2 weeks, and we know that you're gonna grow even more in the future. We know from experience that the bonds that you have created here will last forever, for at least 10 years in our experience, and even more afterwards. And your path has just begun. Now you will always be in the Science Fellows community with all of us. We are around 6,000 alumni from the science education programs of Fundació Catalunya La Pedrera, and there's a lot of opportunities for you to continue connecting with your BIYSC participants, other BIYSC participants, and a lot of talented researchers all over the world. Thank you.
And before we say goodbye, we have one last surprise for all of you. So let's relive some of the best moments of this 10th edition, a very special one with all of you. So now enjoy, and we wish you safe travels, every success in the future, and we hope to see you again very soon. Enjoy.
I really enjoy that you can not only learn about the theory, but then literally just today, the day after learning things, I got to apply it right away. And I enjoy that you can see what you're learning right in front of you, and it's not just on a book.
BIYSC has reached 10 editions, welcoming more than 1,000 students coming from over 70 countries. And every year we continue to grow. This has been the biggest edition yet, bringing together outstanding scientists that engage in such meaningful and immersive research projects.
Because it's an international program, there are people from various different backgrounds, so it's been really interesting to learn about different cultures and different lifestyles.
I think have an open-minded, because you're going to meet a lot of people, a lot of cultures, and also it's going to be a very intensive week.
Really inspired to hear about the 16 research centers coming from different parts of Barcelona.
I learned how to work in a lab, like a real-life experience for possible job opportunities in the future, and learning how to collaborate with my peers in the science fields.
Now I realize that there's a lot more like mechanical engineering, there's a lot more like hands-on practicality that I really do love about science.
It feels like we're all like really connected. Everybody likes each other, and it's like a really positive environment to like learn and like make friends.
During these 2 weeks, the staff team has been the beating heart of BIYSC. They have made this experience truly unforgettable for all participants.
Today we went into an open heart surgery, and that was great because I want to be a cardiovascular surgeon in the future. So seeing how our heart is in real life and actually seeing it live in front of my eyes was a bit crazy, so I enjoyed that.
In school, it's like you learn the content, but a lot of the time there isn't a real-world application, and coming here and seeing the Mare Nostrum, you can see how like people interact and like the careers that people have. And specifically in our course with learning about urban design, we can like apply it to our own cities. I think it's been really informative for like changing my perspective on science.
I've made many great friends that I don't want to say goodbye to when the 2 weeks end. Not only that, but also the project, which I love. We go to the anechoic chamber, to the recording studio, and we also got to see La Pedrera, and also the amazing staff and the activities they do.
I feel like I've really learned a lot, especially like what it is like to work as a researcher. We've been doing like behavioral video analysis, and I found that like really enlightening on what it will truly be like in the future, and it's inspired me to continue my journey to study neuroscience.
I think this program will have a significant impact because it provides challenge way beyond the school curriculum, which, as me personally who loves science, it gives me the challenge, allowing me to move onwards in the maths career, since I'm taking the maths course right now.
My program, and I'm like the people I met their programs, and I'm seeing what they're doing and how much research goes into things, and I learned so much more about the process of it, and I think that it was so interesting to learn that.
I'm definitely like really satisfied with everything I've been learning so far.
I think this connection between staff members and participants is truly special because these staff members have been through the same experiences as the participants. They have participated in BIYSC or in other science programs of Fundació Catalunya La Pedrera, so they really know how this experience feels like from the inside.
I think for them it's like really valuable to see how it's the daily life in a hospital for doctors, because maybe they're considering some science degrees to study in the future, and this really helps to them to decide what they want to do, how they want to do it, and to know how it's the reality like behind the degree.
All the people who are around me share the same passion with me, so I can talk to them about anything around this matter, and I can also build new connections and feel safer basically around here.
I think it's going to have a very big impact on my future and what I choose for college.
I think that for them it may be an opportunity to give them information about real things and real life.
They need some kind of connection to the real world, to the future, to what their future self could be doing. And I think it's very important to get to know people that have been in your position and that have pursued a career in something that you find interesting, but you are not sure how it can end.
I've never really worked in a scientific area where there's so many people who are that experienced. I've only ever worked with teachers. So having that change of mindset really helped me learn more, but also like look into how it would potentially be if I were to work in a research area.
I feel like it's a good way into seeing like my future, right? And seeing like how people before me who have similar interests in like genetics, because we're doing genetics and genomics.
Working with my friends taught me this, and I think I can be able to apply this in the future. It will help me a lot.
One of the things we're most proud of is staying connected with our alumni. We're still in touch with participants from the very first editions, and they are now pursuing their PhDs. They are taking on high impact roles in research and in industry around the globe, and they take the time to reach back, to give advice to current students, to share their experiences, and to inspire the next generation.
I went to BIYSC in 2017 in its second edition.
BIYSC has always been on my mind since then, and being here today as one of the researchers for the guys, it's been really like a full circle moment, and yeah, I'm really glad that I'm here.
10 years in the future, I think I'll probably be wearing some kind of lab coat in some kind of lab somewhere, and I'll be researching around this exact topic, something about regeneration, something about immortality or aging, because I am genuinely very, very interested in it.
BIYSC is a really enriching experience, as I've met people from all parts of the world, and I take a lot of fellows and network that I can do in the future.
Every time that the people that come here to talk to us and to explain things, it's like they know so much about like not only planarians but biology and life, and I feel inspired because I want to be like them.